In this video lecture, part 2, we're going to continue our introduction to genetics and genes. And to start off with, we're going to go through a review of DNA and RNA structure. And I might have to bring up my iPad here to do some annotation on the screen. And as always, please forgive the any writing that looks like a kindergartner's doing it. This kind of technology is still not quite where it needs to be. All right, so we have already in the past talked about it, and/or you have reviewed back in Chapter 1 the different types of organic macromolecules, and we talked about how nucleic acids are your information molecules, DNA and RNA, and those molecules are polymers or chains of nucleotides. So a nucleotide is the smaller organic molecule that serves as a building block for DNA or RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. And the deoxyribo and the ribo part come from the names of the sugars that are components of the nucleotides that make up DNA and RNA. And speaking of that, here is a generic nucleotide. All right, so we reviewed this in Chapter 1. A nucleotide is a building block of DNA or RNA, and a nucleotide itself can be divided up into 3 sections. One is the nitrogenous base, or the nitrogen-containing base. That is linked to a sugar molecule called a pentose sugar because it has 5 carbons in it. And then finally, that sugar is linked over here to what we call a phosphate group, which is a phosphorus atom, abbreviated with a P, surrounded by 4 oxygens. So a nucleotide can be divided up into those 3 sections. All right, and really the most important part from an information standpoint in a nucleotide is what is that nitrogenous base, that nitrogen-containing base. All right, and so in DNA, those bases will look like this. So they're either going to be adenine, which is abbreviated with an A, guanine, abbreviated with a G, cytosine, abbreviated with a C, and thymine, abbreviated with a T. So you can see there, the more detailed chemical structures of A, G, C, and T are drawn out for you. And from an information standpoint, cells treat these almost like letters, like chemical letters. They're going to provide a code or instructions that are going to ultimately lead to the traits that an organism has, believe it or not. All right, now how about, what are the differences between DNA and RNA? Well, first of all, let me point out there is another nitrogenous base called uracil. And if you look carefully there, uracil and thymine look very much alike. But thymines are found in DNA. Uracils are found as a substitute in RNA. Hopefully all that's reviewed for you. Now what about the pentose sugar? In DNA, that pentose sugar looks like this. In RNA, it looks like this. And remember in a lot of these diagrams, like right there and right there, you've got a point. They don't have a letter drawn in. And there are carbons at that location. They're just understood to be there and are not drawn in to help simplify the diagram. But if you notice right there, you have a hydrogen. And right there, you have, that's called a hydroxyl group when you have an oxygen bonded to a hydrogen. So the difference between deoxyribose and ribose is you are missing that oxygen right there. And that's where they get their names, deoxyribose. You have given up the oxygen is missing, and then that's ultimately where these names come from. Deoxyribonucleic acid for deoxyribose, and then ribonucleic acid for ribose, present in the RNA molecules. All right, so your key differences between DNA and RNA. They have those different sugars. You don't have thymine in RNA. Instead, you have uracil. You don't have uracil in DNA. You should have thymine instead. And then generally, DNA molecules are double-stranded. RNA molecules are single-stranded. All right, and we've talked about this before as well. Here is your generic DNA molecule showing its structure. And almost all DNA molecules are double-stranded. There are some cases in viruses where viruses can have single-stranded DNA genomes. When we're thinking about DNA inside cellular living things, DNA molecules are going to be double-stranded. And so how do we build these double-stranded molecules? Well, you've got deoxyribonucleotides. Those are the building blocks that get linked together to build a single DNA strand. And that's what that's going to look like this over here. Let me wipe all that out. There's a very short strand, a single strand of DNA. So you've got a nucleotide right there, you've got a nucleotide right there, and right there. You got an A here, G, T, and C. Those are the nitrogen-containing bases. They're not drawn in for simplicity, but there you can see there's a sugar right there and a phosphate, and the phosphate right here is linked to the next sugar. The phosphate here is bonded to that sugar. The phosphate right there is bonded to that sugar. So we call this the sugar-phosphate backbone. Individual nucleotides within a single DNA strand are held together by sugar-phosphate bonds, and that creates the sugar-phosphate backbone. Now, in a DNA molecule, a double-stranded DNA molecule, you have two of these single strands that are held together through what's known as complementary base pairing. So what that means is an adenine nitrogen-containing base on one strand will always be paired with a thymine over on the other strand. A G on one strand is going to be paired with a C over on the opposite strand. And so you can see that here in our generic DNA molecule diagram. A over here paired with T, G over here paired with C. Notice these little dots in the middle? Those are indicating hydrogen bonding. That's a type of bonding between atoms or molecules where in a typical covalent bonding, when you study that in a chemistry class, you got atoms that are held together through covalent bonding to build molecules, and they're doing that because they share electrons with each other. Hydrogen bonding is a weaker type of bonding where you've got molecules that are sharing hydrogen atoms with each other. So there's a hydrogen that kind of bounces back and forth between adenine and thymine. There are actually two of those, two hydrogen atoms bouncing back and forth. There are three hydrogen atoms that bounce back and forth between guanines and cytosines. And if you look carefully here, they've got two little rows of dots between A's and T's and threes between G's and C's. That's because your A and T base pairs share two hydrogen atoms with each other. Your G's and your C's share three. What that means, too, is G's and C's are actually a little more tightly bonded with each other than the A's and the T's. And another thing I want to point out to you guys on here, all right, here's one DNA strand. The purple represents the sugar phosphate backbone. And over here, here's your other one. And what I want to point out to you guys is, notice they've got this 5 with an apostrophe next to it, and a 3 with an apostrophe there. Those are called the 5 prime, so that apostrophe is pronounced as prime. The 5 prime and 3 prime ends of the molecules. And what that 5 prime and 3 prime, what those are referring to are the carbons in the sugars. So we said those are pentoses. There are 5 carbons in each of those. So there's a carbon right there, and notice that's called your 1 prime carbon. There's a carbon right there that's called the 2 prime. There's a carbon right there called the 3 prime carbon, 4 prime carbon, and then finally, sorry, my iPad's going wonky over here. You can see there's a 5 prime carbon sticking out right there. And so the carbons are numbered. And the 5' end, remember you've got a sugar bonded to a phosphate. So that 5' carbon on this sugar is attached to a phosphate, a phosphorus surrounded by 4 oxygen atoms. And if you've got a linear DNA molecule, so it's not a closed circle like we were talking about with bacteria, but a linear open-ended DNA molecule, a strand is going to have one end where there's a free phosphate. It's not bonded to another nucleotide. Down at the other end, you're going to have a 3' end, and that's actually going to be a hydroxyl group, an oxygen and a hydrogen from that sugar that is not attached to a phosphate that would be coming from the next nucleotide. So we say that they're on a linear DNA molecule, there's a free 5' end where you have a phosphate hanging out there, and then there's a free 3' end where you have part of sugar, this oxygen and hydrogen from a sugar that's not attached to another nucleotide. All right, and so what I want you to notice is on our DNA molecule over here, we got one strand where the 5' end is up here, 3' end is down here. You trace that backbone, that's one strand. And then over here on this one, 5' to 3' is going that way. So your two strands of DNA are always oriented in opposite directions from 5' to 3', and that's an important feature of DNA structure as well. We're not going to get too hung up on 5' and 3' and so forth. You know, for our purposes in this class, you can kind of think about the 5' end as almost like the beginning of the DNA strand and the 3' end as the end of the strand. Same thing with RNA. RNA molecules are going to have 5' and 3' ends as well, but they're typically single-stranded molecules rather than double-stranded. All right, as I mentioned on the last video clip, genome sizes, DNA molecule sizes, those are typically reported in what we call base pairs. So 1A and T is a base pair, GC is another base pair, CG is another one, etc., etc. All right, so the lengths of DNA molecules are typically reported in how many base pairs you have on that DNA molecule or in an entire genome. So the smallest known cellular genome that we know about comes from an organism, Mycoplasma genitalium. I bet you can figure out where that organism lives. That's a type of bacteria. You may remember that we talked a little bit about the mycoplasmas back in Chapter 3 when we were going over bacterial cell walls, because mycoplasmas don't have cell walls. They don't have peptidoglycan like almost all other bacteria do. The Mycoplasma genitalium, its entire genome, a single DNA molecule, 580,070 base pairs. All right, then good old Escherichia coli or E. coli, which lives in everybody's colon, its DNA chromosome, it has one chromosome, it's circular, 4.6 million base pairs in length. And you know, this is interesting too. If you could take that DNA out of one single E. coli cell and stretch it out, it would be 1,600, this is micrometers long. A micrometer is 1/1,000,000th of a meter, and 1,600 of those is actually 1.6 millimeters. That's something you could see with the naked eye. An E. coli cell, though, is far, far, far smaller than that. The length of an E. coli cell is just a few micrometers, yet its DNA, if you could stretch it out, would be 1,600 micrometers. And that, again, just emphasizes how DNA inside cells has to be very tightly wound up and packaged to be able to fit inside that cell. All right, how about the human genome? 3 billion base pairs and 1.6 meters in length. Now that's referring to a set of 23 chromosomes, so chromosome 1 through 22, and then one of your sex chromosomes. 3 billion base pairs, 1.6 meters. A meter is over 3 feet long. And that's the amount of DNA, the length of DNA from one set of your chromosomes in one cell, if you could stretch all that DNA out. But remember, our cells are diploid, so we have 2 of each chromosome. And so the reality is you've got 6 billion base pairs of DNA that are almost 4 meters in length inside one of your cells. So think about that. Some additional terminology, the DNA sequence. All that is, if you have a DNA strand, what order are those nucleotides present in? And what's the AGCT letter code? Another term I want to, again, emphasize with you with DNA and RNA. When you see this word base, that's referring to the nitrogen-containing bases. And a base is going to be A, C, T, G, etc. And so a lot of times you won't hear those referred to as nucleotides because it's that AGCT part of a nucleotide is the nitrogen-containing base, and that's where the information is located. That's what the cell is using as a code. So just some, there's lots of new terminology in genetics that you have to get used to. All right, so I'm going to stop this video lecture here, and we'll continue on with this introductory section in video lecture 3. We're going to talk a little bit about how DNA gets copied. We've talked about DNA being the inheritable molecule that provides all these genetic blueprints responsible for traits. So how does DNA get copied so it can be passed along to offspring? That's called DNA replication, and we'll get into that in the part 3 lecture video clip.